Today I am introducing the nerve muscle physiology topic. This is the introduction of the nerve muscle physiology. Very important topic. And few subtopics will be covered in the examination too. I stress upon them. The learning objectives of today's class is structure and function of a neuron, and neuroglia, then nerve growth factors. This is the structure of a neuron. The entire nervous system is made up of neuron or nerve cell body, nerve cell we call it as, which is the structural and functional unit of the nervous system. It has three components or parts. One is the up to here is called cell body or soma. From here to here it is called your axon or nerve fiber, and the remaining rest of the part is called terminal buttons. The length varies in various regions of the body depending upon the action. And almost all the nerve cell neurons will be similar in structure. The nervous system is made up of nerve cells and also glial cells, which are known as supporting cells to the nervous system. They are not conductive like a neuron. They give physical support to the nerve cells. If you focus the first structure, the nerve cell body or soma or the pericardion of various sizes, various forms like a stellate, round, pyramidal, fusiform, etc. It has plenty of granules in the cytoplasm of the cell body, known as Nissl granules. These are made up of RNA with the proteins. Mainly they are concerned with the synthesis of proteins in the cell body. Along with Nissl granules, neurofibrillae are there, fine thread-like structures, 6 to 10 nanometer in diameter of various lengths. They travel in the cytoplasm. They run through the cytoplasm, plasmic matrix, forming a loose framework of fibrils in the cytoplasm. Dendrites are the processes that are connected with the cell body. These are the processes which are receptive in nature. They receive information from other cell body and sending the information to the cyton. 5 to 7 processes are there, and they leave the cell and they are connecting with the other cell. Even they too contain Nissl granules, mitochondria, even neurofibrillae. Already I mentioned they are receptive in processes of the neuron. Impulse can be transmitted from one dendrite to the another. This is how the information is conveyed to the CNS. The long structure, the second structure is called axon. It's known as axon cylinder or nerve fiber, which is originated from cell body called axon hillock. This region, there are no micelle granules are present. Cytoplasm in this region is known as exoplasm. Like a cell membrane covering a cell, here the membrane, cell membrane covering it is called axolemma. Its length is varying microns to centimeters, 90 centimeters. Its cytoplasmic extension with the specialized function of conducting the impulses away from the cell body. Mainly it is involved in conducting of the information away from the cell body to the other cell. The terminal, the third structure, what I was talking about in the beginning, is known as synaptic knobs, also called synaptic terminal buttons or axon telodendria. They are divided into branches with small knobs, end with the knob called synaptic knob. They contain granules or vesicles which are filled with the neurotransmitter, and till the message reaches them, the neurotransmitter is not released. Once the message reaches to them, it is released into the synaptic synapse, the space between a nerve fiber and the nerve terminal. In the figure, in the first slide, I have shown you the axon or nerve fiber is covered by myelin sheath. At regular intervals there is a space is left. It is not continuously covered. It works like an insulator. Insulator doesn't allow any current to leak from the nerve fiber, having Schwann cells which multiply the myelin sheath and constantly forming concentric layers around the nerve fiber. The spaces between the myelin sheath is called nodes of the Ranvier. Okay. So the process of the development or production of the sheaths around the nerve fiber, myelin sheath deposition around the axon, this process is called myelinogenesis. In CNS, it is done by the oligodendrocytes. Oligodendrocytes are the one sending processes up to 40 axons in the CNS. Here are the myelinogenesis occurs in this fashion. This is a myelinocyte, and myelination is taking place around the axon. This empty area where myelin sheath is absent is called node of the Ranvier. Okay, this is the three structures. Receptive zone is called cyton. Initial segment of the axon, synaptic knob. This is the organization of a neuron, functional organization of the neuron. Now, I am here to talk about the differences between, there are two types of neurons are there. Now, nerve cells are there, or nerves are there, myelinated and unmyelinated nerves. Remember, every time a nerve is different from nerve cell. Nerve cell is a single unit. Nerve is a bundle of nerve cells collectively enclosed in a connective tissue sheath is called nerve. Nerves may have the myelin sheath, are called myelinated nerves, may not have the nerves, they do not have the myelin sheath, are called unmyelinated nerve fibers. Myelinated nerves are the one, they were formed from Schwann cells, whereas unmyelinated nerves, they are simply buried in the Schwann cell. Examples of the myelinated: all preganglionic fibers of ANS, nerve fibers in the somatic nervous system more than 1 micrometer in diameter, all postganglionic fibers of ANS are belonging to unmyelinated fibers, nerve fibers in somatic nervous system less than 1 micrometer in diameter. Myelinated nerves conduct the information very fast, 50 to 100 times faster, because of only the myelination, the known as saltatory conduction. Very, very important for exam. The point to be noted: saltatory conduction. Conduction of nerve impulse is slower in unmyelinated fibers. In detail, we will see later about it. The second component is a glial cells. We also call neuroglia. In the beginning, I told you they are supportive in nature, supporting cells to the CNS. Supporting cells of the nervous system are called neuroglia. I also said neither conduct the action potential nor form the functional synapses with other cells. There are types of glial cells in CNS and PNS are there: astrocytes in fibrous and protoplasmic, oligodendroglia, microglia, epididymal cells are the glial cells in CNS, whereas in PNS, Schwann cells and satellite cells. Here are the three structures of the neuroglia. Microglia, the first one, which is smaller in structure, so-called micro. Astro, like a star-shaped. Oligodendrocyte, we'll see the function of oligodendrocyte in the next slide. Astrocytes are the neuroglia involved in the formation of the blood-brain barrier. Not only that, absorbing excess amount of the neurotransmitter released and preventing its spread to the neighboring neurons and preventing and have got the trophic influence on the neighboring neurons. It also participates in signaling between them in the nervous system, are the functions of the astrocytes. Oligodendrocytes are responsible for myelination in the CNS. Myelination, already I told you, the formation of myelin sheath around the neurons of the CNS. This is the picture of oligodendrocytes. The microglia Phagocytic in nature in the CNS, central nervous system. Ependymal cells and choroidal cells produce the CSF, also participate in the blood CSF barrier. Schwann cells are responsible for myelination in the peripheral nervous system. Okay, both A and B are together called microglia, so this is not important for you. So now the third component of the cell is the nerve growth factor. Growth and maintenance of the sympathetic neurons and some sensory neurons, which are mediated for survival, and suppression of the apoptosis, are called nerve growth factors. Neurotrophin 3, neurotrophin 4 are the example for the nerve growth factors. There are some brain-derived nerve growth factors are also there for survival of existing the neurons, growth and differentiation of the neurons, new neurons in our body. Other than them, other growth factors all belongs to now, they all are not the nerve growth factors. They may be different in function, but they are also growth factors like growth factor, ciliary, neurotrophic factor, glial cell derived neurotrophic factor, insulin-like growth factor, epididymal growth factor, activin, inhibin, placental growth factor, platelet derived growth factor. Many growth factors are there which are similar with that of the neurotrophic structure. So the mechanism of activation of the growth factors includes tyrosine kinase receptor causes dimerization, causing MAP kinase stimulation, causes transcription leading to gene expression is altered. It is cytokines are famous structures, hormone-like substances, lymphocytes, macrophages, neurons and glial cells. The cytokines whose amino acid sequence is known. If their sequence is known, we call them as interleukins. Even interleukins belongs to the growth factors. They are of many subfamily. Under subfamily 1, erythropoietin, colony stimulating factor G, interleukin 4, interleukin 7, growth hormone, prolactin, all comes under subfamily 1. Subfamily 2, interleukin 3, interleukin 5, interleukin 6 and 11, colony stimulating factor GM, CNTF, oncostatin, M, LIF, all belongs to subfamily 2. Interleukin 2, 4, 9, 15, all belongs to subfamily 3. Interleukin 1, the family first non-interferon cytokine to be identified, and hematopoietins, already I mentioned, almost all they required for growth of the cell and angiogenesis. Most of the 2, 7, interleukin 12, 15, EPO, CSFM, CSFG, CSFGM, all are have got their own functions. Interferons family, alpha, beta, gamma, 10, interleukin 10, tumor necrotic factors, alpha, beta, interleukin 17 family, recently discovered. Chemokine family includes complements like C5a, leukotrienes, polypeptides, interleukin 8. So these all are nerve growth factors. The interleukin 1, 6 and nerve necrotic factor, tumor necrotic factor alpha, these are involved in the causation of the fever. This information we already know. So this is about some information about nerve growth factors, and in detailed analysis and understanding of the nerve growth factors is still required. With this, I have concluded my lecture of Introduction to Nerve Muscle Physiology is done. Hope you understood my class. Any doubts are there, please comment in the comment section. If you like my channel, please subscribe it and pass this information to others. Thank you so much.